Thank you, Aris, and also just to recognise, I think there's a very strong collaborative working relationship that's been developed between Biobank and Regeneron through the course of this project, and certainly to recognise the contributions of particular individuals, so kind of John Overton, Jeffrey Reed, John Penn, and the many others in Regeneron who are really contributing to the success of what is quite a complex delivery program to achieve. So we've talked about genotyping, we've talked about exome sequencing, and so I'd like to welcome to the stage Professor Nicole Soranzo, who is the head of the Human Complex Traits Group at Wellcome Sanger Institute, and a member of UK Biobank's whole genome sequencing expert working group, to share some of the perspectives on the whole genome sequencing program that's about to start.
So good morning, everybody. So mine will be a very data-poor talk. So I'd like to give you some perspective about how we're thinking about whole genome sequencing in UK Biobank. It seems really hard to think that less than a year ago we met with a group of investigators at the Wellcome Trust to discuss this prospective project, and the project indeed already started. And this is really a testament to the ability of UK Biobank and our funders to really buy into the vision and get things done. The Genome Sequencing Project is part of the UK government's Industrial Strategy Big Challenge Fund for and really has the ambitious aim to produce a deep sequence characterization of the whole genome sequencing of the entire UK Biobank. So there are many logistics still to be, or if you want details, still to be worked out in terms of both scientific, technological, and organizational details, but really the ambition is to complete the sequencing of the entire cohort within the next 5 years. And the work has been divided into two main stages, so a pilot or vanguard phase, which will sequence the first 50,000 individuals, and then the main phase, which will sequence the remaining 450,000 participants. And indeed, as I mentioned, the vanguard phase has now officially started. It will sequence 50,000 participants using Illumina short-read technology. For those of you who like sequencing, this will be done using 151 base pair paired and PCR-free libraries. We aim to achieve a 30x average coverage of the human genome with 85 gigabases minimum per genome. The sequencing will be carried out at the Sanger Institute and will start officially the 31st of July of this year. And the plan is to have the entire 50,000 individuals turn around for sequencing in the next 18 months. And just to give you an idea of the scale of the challenge, so this is predicted to generate about 4.5 petabases of sequence, which is pretty close to the entire sequencing output of the Sanger Institute until now. So it's really a major scale up for us as well. And in addition to this project, we will also be running a series of innovation projects, as Mark pilots has already alluded to, which will really inform technology for the main phase of the project. So I really want to stress that this is in no way a competitive effort to the exome, but rather builds on and is really complementary to both the genotyping and exome sequencing data sets. And I thought it would be really worth it to think about what kind of information or data we will get out of the genome sequencing project. So this plot is depicting the proportion of single nucleotide variants that is indeed informed by an empirical evaluation study where we had the same individual sequence with exome sequencing of 50x, genome sequencing, and imputation. This is really an illustrative cartoon which represents a lower bound estimate. But the main point here is that using a standard traditional exome capture array, you are only able to see between 2 and 2.5% of the total variation that we see from whole genomes. And we know that much of this variation is actually not accessible to imputation. Even with latest and more advanced imputation reference panel, we know that both the percentage of SNPs that can be recovered and also the accuracy of the imputation falls quite dramatically below 1% minor allele frequency. So that indeed the vast majority of variants below this threshold are not imputable. So what the genome sequence will give us is access to the remaining 98% or so of the human genome, and also access to the hard-to-impute fraction of human genetic variants. So obviously, the first thing we're interested in understanding is what is the contribution of these rare variants to human phenotype. And you heard very well about the value of coding variation. But the question is there equivalent scope and value to look outside protein coding regions? We think this is true, this is the case. And one of the studies that informed this thinking was actually based on the phase 1 of the UK Biobank imputation. So this was published about 18 months ago by my group with many collaborators. And here we carried out a whole genome-based scan in about 170,000 individuals, looking at 36 phenotypes that capture variation in full blood cell counts. And so what we found is the largest to-date number of independent variants with 2.7 thousand statistically independent associated variants. And what was quite remarkable is that for the first time we discovered a large number of what we call low frequency, between 1 and 5%, or rare genetic variants. And so this really allows us to study the properties of these variants in bulk. And so what you see here, as this is the allelic spectrum, is that indeed the first observation is that there was really a continuous of genetic effects between the common low effect variant, which we discover in typical genome-wide association studies, and indeed variants that are low in frequency but begin to approach effect sizes that are comparable to clinically actionable variants. And indeed, what was even more remarkable, if we divide these variants based on where in the genome they fall, we see that for common variants, as we would predict, the vast majority sit outside protein coding region, as highlighted from this yellow, blue, and green color. But indeed, nearly 70% of the rare variant associations also map outside protein coding genes. And what is even more striking is that if you plotted the average effect size for the association, this rare predicted non-coding variant have similar effect sizes to rare, highly damaging coding mutation and much greater than the common counterparts. And this really suggested that there is potentially a pool of clinically very informative genetic variants that are outside protein coding regions that we want to discover. Also, this information comes from other studies, and this is one by Matt Hulse, based on a DDD study, which looked at children that with developmental disorders that are undiagnosed based on all exome sequencing studies. And what they were able to find is that there is indeed a high rate of de novo mutation that are mappable to ultra-conserved elements in this case, of a relevant tissue type, which is a fetal brain. And so this really suggests that a deep understanding of the genome structure, including mutation rates, demography, and natural selection, coupled with really deep regulatory annotation and also with a growing number of large-scale experimental study, will really allow us to identify for each disease non-coding region, particularly regulatory regions that are of particular interest for specific traits on disease. And so we can think of then looking at association of rare variants under this region in a manner similar to what we're doing now for coding variants in exomes. And we done a few power calculations just to understand what spectrum of effect sizes we think we can capture. So in this case, we test association with aggregated rest of rare variants, where by modeling different cumulative effect sizes for the combination of these genetic variants and their cumulative allele frequency. So in the 50,000 of the Vanguard study, we have a strong power to detect association in this top right part of the corner. And just to give you an example, for instance, we have full, nearly perfect power to discover association with a cumulative effect size of greater than 0.6 standard deviation for variants that are found in 6 people out of 1,000. And in the full data set, we obviously have more power. And so for the same effect sizes, we will be able to detect effect when there is at least 4 carriers out of 10,000 individuals. So we really think this is going to be a very worthwhile effort as our understanding on the non-coding part of the genome improves to really discover many more associations. So the other thing I wanted to point is that indeed sequence variation is only a very small part of the overall picture, what makes us biologically interesting creatures. And indeed, the genome sequence will really allow us to look at very diverse classes of genomic variation. The perhaps most important one are different classes of structural variation. So there are now available software that use properties of short-read sequencing to derive information of many classes of variation, such as deletion, duplication, inversion, and mobile element insertion. And it is very clear that genome sequencing will foster the development of even more accurate ways to predict and study this variant, and particularly to evaluate their contribution to human phenotype. The other interesting aspect of biology is telomere length. It's been shown by Richard Durbin and colleagues that indeed you can use now computational methods to derive quantitative measurement of the length of telomeres, which is indeed very well correlated to traditional methods such as Southern blot. And so this really opens up a possibility of beginning to really study and understand the role of telomere length variation, both in basic biological processes such as aging, but also in complex diseases, as shown in this case, in this example for types of cancer. And a third aspect, which is also, I think, going to be really important, is our ability to study mitochondrial DNA variation and its impact on human traits. So mitochondria are really humble, small molecules with 17 kilobases in humans. They have only 13 protein coding genes. They are mostly implicated in electron transport chain, which in turn control OXPHOS function, as well as a few number of T and rRNA that control the synthesis or the translation of MT-encoded proteins. And so we know already that mitochondrial function is governed by both mitochondrial variants, nuclear variants, and environmental factors. And indeed, this function, which is also typically associated with aging, leads a progressive bioenergetic decline that has been correlated with many diseases, including immunological, metabolic, and degenerative diseases, but also with cancers and aging. And so we will be able to use genome-wide sequencing to study this. So we know already that indeed from array data we can call very accurately mitochondrial haplogroups, which show in Europe this structure function. But what the sequence will give us is the ability to resolve causal effects by resolving effectively variants that define sub-haplogroups with different functional properties. And another interesting aspect of mitochondrial biology is indeed copy number. Copy number variation of mitochondrial has been associated with tissue-specific variation, which has been linked to stress, and indeed has been associated now with clinical endpoints such as major depression. So the mitochondria has about 100-fold coverage compared to autosomes. And so we'll be able to very accurately call mitochondrial DNA variation from our genome sequencing data and then correlate it to a host of phenotypes and disease outcomes. So this is really to conclude by saying that indeed from the sequencing data we will be able to achieve a broad exploration of different classes of genetic variation, including rare encoding regulatory variants, structural variation, telomere length, mitochondrial DNA. It's undoubtedly the case that this will produce many useful tools for genome analysis and not unimportantly also an enhanced imputation reference panel that can be extended to other disease data sets. And this will allow us to really address many very fundamental and broad questions about biology, and these are just some of the examples, around the contribution of rare variants to complex traits and application, for instance, prediction, as you heard before, understanding of the role of regulatory variation and really deciphering the regulatory grammar of the human genome, ability to really map with high certainty in the landscape of evolutionary and mutational constraints in the human genome, and this really provides us with a background information onto which we can overlay disease-causing variants. And finally, and importantly, phenotypic processes that are associated with not only this level of variation, but human evolutionary processes at large. And really, I joined the chorus of people saying that this really will empower our UK and worldwide science. And thanks very much again to the UK Biobank for making this possible.